Good morning and good afternoon, everyone. My name is Samantha McClenahan, and I'm a program lead here at the Digital Medicine Society. It's a pleasure to welcome you to our launch event for our initiative focused on advancing the development of risk prediction for cytokine release syndrome through leveraging digital health technologies. We're thrilled to have you join us for this exciting session. We have an action-packed agenda ahead of us where we'll introduce our resources developed through this pre-competitive collaboration held by DATAcc by DIME. We'll also dive deeper into their impact and the success of this collaboration through panel discussions with experts from our project team, as well as a fireside chat with a special guest speaker who has personally experienced some of the challenges with CRS. But before we begin, we need to cover a little housekeeping. First of all, we are recording today's session. That's great news for you. You'll be able to access these slides and the DIME website following this session to share with your colleagues and to continue the discussion. If you have any questions along the way, please send them through the chat. We'll be monitoring, and if we can get to them, we'd love to. We'll also be dropping some links along the way so you can explore the resources and some of our case examples. And finally, please do not transcribe this webinar. Before we jump into today's discussion, I'd like to share a little bit about our organization. The Digital Medicine Society, or DIME, is a global nonprofit that sits at the intersection of healthcare and technology. We leverage our growing community of experts to tackle some of the most complex issues in clinical care and clinical research. Our mission is to advance the safe, effective, and equitable use of digital approaches to redefine healthcare and improve lives. We achieve this through research, education, and tracking best practices. By harnessing the digitization of healthcare, we aim to tackle some of the most persistent and urgent challenges facing our healthcare system. One area where digital technologies can make a profound difference in enhancing the monitoring and management of drug-related adverse events. With the growing availability of DHTs and their continuous evolution, we now have the opportunity to more effectively monitor and track treatment-related risks during clinical research and clinical care, ultimately ensuring safer outcomes for patients. This brings us to the heart of our project, which recognizes both the immense promise of emerging immunotherapies such as CAR T-cell therapy and bispecific antibodies, and the common significant risk they pose, cytokine release syndrome, or CRS. CRS is a potentially life-threatening side effect of some of these immunotherapies, characterized by the rapid release of cytokines into the blood by immune cells affected by this therapy. The risk of CRS often requires regular clinical oversight through extended hospitalization, creating barriers to care, increasing financial burdens, and limiting patients' access to these vital immunotherapies. To address these challenges, about a year ago, we brought together a team of experts to explore how digital innovation could be harnessed to better understand and mitigate the impact of CRS to make these immunotherapies more accessible. And through this project, we have shared, collaborated, and grown together to identify what are the early digital clinical measures of CRS and how can we standardize the collection of those measures to build robust data sets critical for developing more performative, predictive approaches, including AI-based models. And to explore what are the technical, operational, and regulatory requirements we must consider as we embark on this journey. I would like to extend a special thanks to the 23 partner organizations shown here on their slide for their dedication and commitment to the continuation of CRS-related projects to de-risk and mitigate their effects. And with that, today, we are excited to share these open access resources with you that are now available on our DATAcc by DIME website. For those of you less familiar with CRS, we provide a brief background on CRS, including some of the current clinical and logistical challenges, before providing you with the most fundamental resource right up front, so you can immediately begin to explore how to build trustworthy CRS risk prediction products. This practical interactive guide offers recommendations for designing and developing evidence-based DHTs for CRS. It outlines the value proposition, provides actionable insight for navigating key developmental milestones in bringing CRS de-risking products to market, and provides support for harmonizing data collection efforts. From this guide, you can navigate to more detailed project resources. And so let's jump in to the early measures for CRS. One of our main project goals was to explore how sensor-based digital health technologies can advance the field by enabling earlier detection through continuous monitoring and supporting the development of CRS de-risking products. To accomplish this, we first needed to identify what we needed to measure. We conducted a scoping literature review to identify early CRS-related measures. And we didn't limit these only to digital measures because we wanted to keep an open mind of the future possibilities that may exist as further exploration of these measures dive in deeper, such as those with physical activity that might occur when we have more data around them, or for the development of new sensor-based digital technologies that might be able to capture objective-based measures or those laboratory measures that we can't collect in the outpatient setting in a more continuous manner. Through these collaborations with our subject matter experts and consulting with key opinion leaders in the field, we also selected a common set of essential measures that can be easily monitored digitally for effective data collection, analysis, and algorithm development. These measures align with some of the current grading scales of CRS and can be passively and continuously captured in some areas, which offers immense potential for real-time predictive modeling. With this data in hand, we now turn to the next step. How can we move from measurements to actionable solutions? Our innovation roadmap provides this framework for developing advanced digital products that can help us lead to reducing the risk of CRS. While DHTs are already available to support CRS monitoring in that outpatient setting, a key question still remains: how can we enhance risk prediction, enabling us to foresee CRS before it develops or progresses into severe cases, allowing our patients to return home or further away from the clinic, and reducing that length of those hospital stays and ultimately easing that clinical burden. We've outlined a strategic path that delivers increasing value for patients and healthcare providers at each step, laying the foundation for more effective risk prediction models and products in the near future. While it's possible to jump in at any stage along this roadmap and develop a product, we wanted to highlight the value of the iterative approach. Each step, we give a little bit of background of what the goal of these products would do, their impacts on patients and the clinical space, and then a little bit of the barriers that we must overcome to be able to see these have success within the field. You can interact with this map, or you can circle back to the guide and explore some of our case studies and other resources on our website, all available here to support you in taking your next step and leveraging digital innovations to de-risk CRS. And now I'd like to turn our conversation over to our panelists today to discuss how these resources will have an impact in the field and how this collaborative community has facilitated this progress. We'll have 4 panel sessions today, and I'd like to start off with our lived experience guest, Michael Hibbert. Michael?
Hello.
It's such a pleasure to have you with us today. We're looking forward to learning from your journey. Could you start by sharing a little bit about yourself and walk us through your experience with CAR T-cell therapy?
Yes, well, I'm, hello everybody, Michael Hibbert, as she said. Well, I've been on a 10-year journey now. I'm a father of 4, a career man, been at the company I'm at for about 20 years now, and they've been a big part of that. I did, multiple therapies along the way. Started out with, I believe the Cyborg D was my first, went into a stem cell, and spent several years, I think just over 5 to be exact, on a very successful clinical trial, which is I think ELO, carfilzomib, if you will. Please correct me, guys. Dexamethasone and the lovely Revlimid. It was about 5.5 years, and that really went good and really went strong. The initial hit was very, very successful. And as this multiple myeloma, it creeps back up, but it done really, really good for a long time. And then eventually I was able to, as that kind of went south on a similar year, we were able to be part of the CAR T-cell therapy here. And that was really, really exciting on my end. When I first started this journey, I remember being part of watching as a show on HBO called Vice. It's a documentary series, and the gentleman there, the producer, his mother and mother-in-law both developed cancer. One was breast cancer, the other multiple myeloma. And he went on the series there of treatments and everything, because at the time, treatments were still a struggle with multiple myeloma, and a gentleman at my age was fairly rare with my type, so everything was still out. And at that point in time, we got to hear a little bit about the T-CAR cell therapy, which at the time wasn't really big with multiple myeloma. It was in other areas, but there was still the hope, and it was real exciting. and obviously that went down, and I know it was kind of up and down with that. And then this past year, with the recent downfall of the clinical trial, Dr. Varga presented me that with a possibility. At first, we was going to hit it, and I was missing some prereqs because I had been on the clinical trial for so long. As Dr. Varga explained, there's been a lot of good drugs that had come on in the years while I was in the closet having good times on my clinical trial there. So there were some prereqs, and we were able to get started on one of the prereqs that had become the new drug now to get it going. And then in the midst of that time, and so I hadn't done that, we kind of had looked away from the T-cell therapy. And then all in the middle of that, things progressed, and she was able to come back and tell me that they had put the T-CAR therapy as a second defense, and we was already able to start the prereqs and all. So that had me in a really good position to do something that I've been hoping I'd be a part of. And then for it to be as successful as it was, as I just heard, thank you, Dr. Varga, a couple of weeks ago, it was very successful, and my body has seemed to react very, very well.
That's so exciting. So great to hear your experience and your background with how you have progressed to this journey. And you really were looking forward to that CAR T-cell therapy. And it was a bit able to come full circle and around a little bit sooner for you. And it seems like you're having a great response. That's such great news to hear. I'd like to dive in a little bit deeper. CRS is a common adverse event following CAR T-cell therapy. Were you concerned about this potential adverse event before you began? And what was your experience like with CRS?
Definitely there was a concern. During the instructions portion of it, I was working with a nurse that was on the very front end of the development there in the area and all, and she was explaining how the CRS syndrome was very intense. I think she might have described it as it's almost like some people kind of went crazy for a moment and all. And so she was telling me how that, and also telling me how, as they progress and all, seeing the side effects and then be able to beat the side effects with prereqs, if I can continue to use that word there, to get everything in control. So yeah, I was going into it, I was a little concerned about how my body would react. I'm an odds guy too. My body's reacted so well to some of this. I figure when my time's come up, to have a really good time with some of these treatments and all. But my body seemed to be, I was concerned going into it, but my body seemed to respond well. I think throughout the first week and all was kind of, it was a vacation at Hotel Hospital, right? Wasn't much action going on. And it responded well. And then at that point in time, I was able, they seen how things were going. I was able to qualify for a home health program. So I went into that and was able to go home, and I was able to be at home, and I had help at home too. My fiance's in-laws, mother and father, my in-laws was able to come up and stay with us. So I was able to go home, have good care, and then obviously with the home health at home program, definitely was great, and it was so good to do that at home. I thought I was kind of unscathed with it all. I was going to ride through it and it'd just be a good journey. Right about, I want to say, right about close to day 10, I did start creeping up with the fatigue, and then I did do probably about a day and a half or so with some high fever spikes. Got consistent, stayed above 101, and then sometimes dipped up close. I want to say maybe close to 103, 104 and all during that process. I was able to manage it well. I am a man sick guy, high tolerance of pain and stuff. So I probably took it too long, and the equipment stuff that was brought along with it that was self-monitored, and we had nurses that monitor you 24 hours a day. And I did receive the phone calls in the middle of the night concerned about my fever. And at that time I was downplaying a little bit, the man sick type guy, and she was, hey, no, this is real important. I remembered how important that was, so they was able to advise me, and we was able to monitor it. It didn't peak really high, but after about a day or so it started coming back down, and my body was able to respond and get everything in control, and able to move past that. Fatigue stayed with me just a little bit for a few more weeks on after, but I responded well.
Awesome. Thank you so much for sharing those very personal experiences. It's been great to hear so many great impacts about that story, from, being able to return home and have those therapies or those additional technologies really be able to help you and allow you to experience that more in the comfort of your home with your support system. So so glad that you had that elements there. They were able to, have a little bit of oversight with, being able to interact with you where maybe you wouldn't have called in and had that. So, so happy to hear how that journey has led you. And we're so glad that you were able to join us today. And thank you for opening up our great discussion. We'll continue now with our next panelist. And so I'd like to think about how we dive a little bit more into the clinical management of CRS and explore more about kind of what Michael was sharing about using DHTs to shape the future of the care for CRS in this area. With that, I'd like to welcome Cindy from Atrium Health, Matt from Best Buy, and Nazila from Johns Hopkins. As we're considering the complexities of CRS, thank you all for joining us. One issue is the potential for it to escalate very quickly. To begin, can we start with thinking about what the biggest challenges might be today in detection for CRS and which clinical signs are often missed till it's too late?
Yeah, sure. I could speak to that. Traditionally patients who receive T-cell redirecting therapy are monitored, in the inpatient setting. We have their vitals taken every, 4 to 6 hours to monitor for any signs of fever or drop in blood pressure or drop in oxygen saturation. But I think the challenge is that some of these patients can have these subtle changes that we might be missing by, checking them, every 4 hours rather than having, for example, a 24-hour monitoring where we might catch things a little bit earlier. I think, the subtle changes that we're looking at are in temperature. Even if a patient's not having a full-fledged fever, if they're having, low-grade fever or even just a change in their baseline, that could even signal that there's a CRS coming. Subtle changes again in heart rate or, even in symptoms of like malaise or, bony pain or muscle aches. I mean, these are all really early signs that we might miss, if we're only checking on the patient every 4 hours or so. So, that's been at least my experience versus when we were monitoring them on like wearables for 24/7. So, I don't know. I'd love to hear other people on the panel on what they think and if they agree.
Yeah, I think that the most concerning symptom for us is hypotension, and that's the thing that makes it an emergency. Like Cindy alluded to, fever is going to be the first symptom. So in a way, catching that earliest is the most helpful because it gives us the most lead time, given the first fever will happen before the first low blood pressure does.
Thanks for jumping on some of those clinical measures. Let's dive a little bit deeper in there. So when we're thinking about those measures that might be predictive of that early onset, how would the ability to predict CRS at that early stage impact clinical decision-making and some of these patient outcomes?
Again, I defer to the clinicians in the room on this one.
Yeah, I think, at least in my practice, if I have a patient who is on a product, got a CAR T product, where I know that the onset of CRS is going to be day 7, and they're starting to have signs of CRS very early on. To me, that's like a big red flag that, we need to probably, intervene earlier rather than later. So sometimes, if I'm worried this patient's fevering too early on, I might start tocilizumab early on. I might start dexamethasone early on, just so I can stop that escalation, because sometimes that's the threshold where they can go into, severe CRS or even have other side effects such as HLH, where you're requiring huge doses of steroids. So if I can intervene early on and prevent that escalation, I would love to do so. So finding signs of CRS that are occurring before they should, for me, is a sign that I have to intervene.
I think in our case, we operate in a day hospital system. So patients come in, get their treatments, are monitored daily for between 15 minutes and 12 hours. And that's great, but they still have to live within 1 hour of the hospital. And given the radius patients travel to get to us, upwards of 6-8 hours by car, what that means is they have to often use a big portion of their life savings to get housing nearby. So what it would mean to be able to detect early is that as long as they have a reliable caregiver, they could get their treatment at home 4 hours away. And if we are able to detect early, then we can give them a nudge to come in and make it to our hospital, which our safety outcomes show, specifically with our bispecifics and chimeric antigen receptor T cell therapies, has a big difference in outcome if they go to a local hospital versus if they come to their tertiary treatment center, quaternary treatment center to us.
Perfect. Thanks for that insight. What about when we're thinking about managing caseloads as these therapies expand in their indications of use and more come to market?
Yes, we are. The volume is definitely going up. I think that the strain on beds in the hospital is a real thing, and so I think we are turning more and more towards remote patient monitoring. And at our institute, we've been doing that for bispecific antibodies. Michael had mentioned the hospital home program, so we have a hospital home program that monitors 24/7 our patients, and they interact with a nurse that's available and an internist who's trained in bispecifics. But we also now have an outpatient cellular therapy program where patients come in every day after CAR T therapy, and they can get tocilizumab, they can get IV fluid, they can get dexamethasone if they need to. So we're really making that transition now towards that outpatient. I think to your question that you had posed earlier, a risk prediction model would also be very helpful when we're considering which patients should do this outpatient and which should do this inpatient. If we had a way of knowing that this patient's at risk of having an untoward complication, I think we would definitely want to put them inpatient. So these kind of risk prediction tools are super important for us.
I think as a lot of you have heard me talk about people on the panel, I am a part of the working group that plans our bispecifics administration and quality improvement at Hopkins, and we have had a lot of difficulty now that there's the first mainstream solid tumor bispecific with tarlatamab, just from a capacity constraint perspective, and that's only going to be exaggerated over time. So I think as Cindy again alluded to, anything that can move these treatments into the outpatient setting and leverage the space we have without compromising safety is huge, because the reality is, at least for us, and Cindy, I imagine it's the same for you, it's not the high socioeconomic, high health literacy patients who have bad outcomes or who tend to present quite late. It's people who don't have as reliable caregivers, don't have as high health literacy, because they tend to wait and then present when they're quite a bit sicker. So I think having a safety net for those patients is huge, and that's what a lot of this remote monitoring would afford us.
Thanks for those great clinical insights. Matt, I'm going to get to you next. What are some more of these specific lessons that we might learn that Cindy has alluded to, maybe through clinical trials or real world experience that shapes some of the approaches of how we're managing CRS?
Thanks, Sam, and thanks for having me. Actually, I was very happy to keep listening to Cindy and Naz. I learn a lot, and thank you to you, Mr. Hibbert. So I really enjoyed your story as well. So I think what's interesting is that it takes a while. I'm an anesthesiologist by training, not a hematologist, so I'm even more obsessed with monitors and real-time feedback, and it takes a little while as a clinician to become comfortable with the fact that you just can't see the person in front of you. And that's kind of the point. That's the therapeutic intent of having people in the home. But it does mean that you don't bug them, but it does mean you have to work out ways to deal with this new data stream that looks superficially familiar, but actually might be a little bit different. And the primary reason why it's different is because you have the effects of somebody's activities of daily living. It's what you want. You want them to go upstairs. You want them to get dressed. You want them to stay moving. You want them to avoid a VTE. You want them to avoid infections. All of this is great. But it means that data becomes more sparse, a little bit noisier, and we have to kind of understand that. So to your question, in both the trials we've supported and the clinical deployments that Cindy's alluding to, there's a big kind of period where everyone gets used to this new stream of data. And I think there's two things that I'd probably call out specifically. The first is that when we think about monitoring in the hospital, we tend to think about patients who are kind of the ideal patient. They might be kind of sitting up, awake, still, and it means that monitors can be clunky and wired and nurse operated and all of these things because you're not relying on the patient to do it for you. So when you then think about the home, you have to think about making monitoring more acceptable, and often that makes it either less accurate because it's dealing with noisier data, or it means coverage is more sparse. And we just have to get used to that and think not that I need 100% perfect monitoring. It's like, what monitoring is enough? And equally, when you then think about how you alarm on stuff, you don't want to bug people all the time. You want to start bringing in specificity. You don't want a monitor that's just purely sensitive and goes bing. You want some means of thinking, is there a chance there's a false positive? How can I eliminate that as much as possible? And that can be done through good alarm setting, but also through human and, to a lesser extent, ML and AI triage.
Thomas, can
Indeed.
you answer your question that you put in the chat? Sam, is that okay?
Yep, go ahead.
How often do patients with who get a temp spike not end up developing CRS or have false negatives? I think it's very relevant for what we're talking about, but I will say if you got CAR T or a bispecific T-cell engager and you have a fever, we just like call it CRS. So I think unless you got it within 10 minutes of getting a transfusion and then it just goes away and then we may be attributed to the transfusion, but I think that points to a greater underlying topic here, which is like what actually matters with remote monitoring, what's the most important, and it's not necessarily that you had grade 1 CRS because so many patients get it. It's the higher grade CRS. That's what's most clinically helpful, I think.
And actually, to sort of zoom out one more step from that, Nazira, it's what we really care about is where does this person need to be? So when I talk about specificity, I'm talking about essentially, are these physiological changes real? I'm not talking about the specific diagnosis of CRS versus, for example, sepsis. What we really care is, do we need to wake up this patient in the middle of the night, and do they need to come to your and Cindy's care for a workup to rule out all the things it could be?
And it's also important to note that at night when we sleep, our body temperature might go up a little bit as well. So those warnings or those alarms really need to also be adjusted for nighttime. So you don't want all those false alarms at night where patients are waking up because of fevers when this is just core body temperature going up a little bit as they sleep. So these are all important things to factor in.
That's actually a lovely sort of minor segue into Arsh's point, actually, which is what kind of temperature is important. And
right.
You're absolutely right, is we treat temperature as this one thing, but actually you've got your core temperature measured perfectly by a pulmonary artery catheter, which may not be what we want to send people home with. And you've got skin temperature in random patches of skin that's a function of perfusion as much as it is a function of core temperature. And it's finding something that is acceptable to the patient, but sufficiently accurate that we can make the diagnosis that we need to make.
Perfect. Thank you guys for that insight. You have led us beautifully into our next panel discussion, where we'll dive more into those technical and performance features of DHTs and the development of these risk prediction products. So I want to thank you all for your insight. And we will now move to our next panel. I would like to invite our panel speakers here, Eric from Takeda, Matt from Blue Spark Technologies, Michael from Actigraph, and Nunzio from Pfizer. Thank you all for being here. Super excited to continue this discussion on those technical features that we were alluding to at the end of that conversation there. I would like to begin by really just diving first into the features of DHTs that are available to use for this continuous monitoring that can capture those similar signals that they're collecting discretely and remotely in clinical settings to maybe more of those continuous streams like Matt was alluding to, and that we were ending out that conversation with. Matt, can I start with you?
Yeah, good morning. Great to talk to everybody this morning. Yeah, I think the really encouraging thing is that DHTs do exist today, digital health technologies, and are showing promise in practical use today. So this is not just some future tech. It's stuff that can be implemented and integrated today. A lot of talk about temperature. We actually happen to make a continuous temperature monitor. We monitor axillary temperature, which is shown to be correlated in agreement with core body temperature. So, as that was pointed out, that is a leading indicator of early onset of CRS. I think there's a lot of good tools to monitor some of the additional vital signs that we talked about here today. I think as we take a look at DHTs, As Michael, thanks for sharing your journey, pointed out, guys, I include myself in that category, they tend to wait till the last minute before they admit that something's really gone wrong. I've done that personally, and I don't recommend it. But the interesting thing about DHTs, they have the promise, one, they have to be easy to use, right? If patients don't use them. Today they'll send somebody home with a stick thermometer, for instance, say, hey, go monitor your temperature every 4 hours. Patient compliance with anything is usually pretty awful, as most docs will agree, whether that's meds or doing that. DHTs, if they're easy to use and for instance, they're wearable, takes the patient compliance out of that because it's automatic. Those signals can come through in an automated basis, a continuous basis, and really allow you for that safety factor, one for the patient, but also it enables them to get into the outpatient setting as well. So it's really a win-win for everyone. As we get further adoption of those technologies, that leads us into, a lot of the work that the group did is into feeding that data into predictive algorithms that can even indicate those factors even earlier.
I think Matt, you mentioned two great points that are related to accuracy and compliance. And when it comes to the selection of a digital technology for a CRS de-risking product, accuracy is paramount. The measurements should be validated against standard of care reference methods, also under different conditions that can simulate the physiological variability observed during CRS. But that's not only the only aspect that matters. There are some hardware specifications that should be matched as well. For example, long battery life, large buffer memory, fast and secure data transfer, and multimodality is also a key aspect. It's important to give priority to those devices that can embed multiple sensors simultaneously, so they can reduce the burden of participants of wearing multiple devices, facilitating data integration, and ultimately providing a comprehensive picture of patient health status. And also form factor is really relevant in the selection. It's important to pick a good form factor between like wristwatches or lumbar belts or chest patches, rings, and so on, because the form factor impacts accuracy and data quality, but it also affect the comfort that in turn, as you mentioned, impact compliance, and we want to maximize compliance. So devices should be non-invasive, easy to use, and they should be comfortable to wear. Comfort is a very crucial aspect, and I think it should be measured both with self-reported measures of usability and by objective measures of the level of continued usage of the device.
Yeah. And I think Nunzio really, I love the phrase he used, a more continuous, a more complete picture. And that's come up throughout the first couple of sessions here. Right now, the standard of care is episodic, right? So we're getting one measure every 4 to 6 hours. Individuals there's intrapersonal variation, right? I mean, my heart rate is low when I sleep. People are wondering what's going on with me. And really, we need to capture that intrapersonal variation at baseline and understand how a person's doing. And then when we see deviations from that baseline post-infusion, that's going to give us a better understanding that something may be going wrong here. And that's only going to happen with a more continuous picture. So it's with that continuous picture that we're able to understand how an individual functions and and what is a meaningful temperature deviation. Is the heart rate spiking, a sign of pyrexia and CRS? Those are the types of things, that DHTs offer.
I think one thing to consider is the foundational data collection piece too, right? It's foundational to where, Samantha, you showed the journey of where we are as evidence generation is important now. It's still early. Matt, you mentioned that there are devices here people are using. We should be using them more. So if we can build upon that to get to that point of where it becomes more of a digital biomarker, being more accepted. But with that, you think about data transfer, specifications, how it's done, how it's harmonized is really foundational. Where we are at this time and space, to be able to build upon that. If we want to get to that point of a product, a useful product, further use outside of the hospital, it's vitally important that there is those standards in place that we can build upon for further downstream modeling and analysis to actually get to the prediction part or aspect of it.
Yeah, thank you guys for that so much. Matt, you kicked us off well with thinking about that continuous data. Nunzio, you hit on those 4 factors that we need to be considering now as we're deploying these DHTs, as well as moving into these more predictive-based models. And then Michael and Eric, you led us beautifully into my next question, which is more focused around how do we think about taking that data and collecting it, making sure it's in the form that can be used, it's accurate and ready to validate CRS de-risking products. And what are some of the challenges that our developers are facing in this space when you're thinking about validating those products for CRS? And what clinical evidence do we need to make sure that they're going to be trustworthy and reliable?
Sure, I can kick off maybe. So I guess when we've been talking to clients, like there is very necessary focus, and regulatory will probably get into this much more deeply, but surrounding the intended use of a device. Like so, I mean, we talk about CRS as a univariate construct here. I mean, obviously a graded construct, but basically something that's very univariate. But there are open questions surrounding like how much of a single variable it is. Like, you know, how fast, so when we're talking about spiking temperature, is it going to spike more quickly in certain indications than others? Is like hypotension much more likely to be in a different place than others? So we really need to think about I guess what we want to target. If it's CRS grade greater than or equal to 1, for example, like, you know, temperature may be paramount. If it's CRS grade greater than or equal to 2, like we may need, you know, we're very likely going to need to think about blood pressure, hypoxia, you know, and measuring those parameters. And we also need to think about how that's being adjudicated. So or graded. So there may be some level of, you know, consideration about the outcome, like and how we're capturing that. So it may be standard in certain settings for CRS to be graded at a daily level. But if the indication for use is to, or, you know, if we're trying to take patients outpatient, and we say we need to beat sort of the standard of care by 2 hours to comfortably move them outside that setting, we need to capture the time at which that's onsetting. So when is that standard onset time? Because if we don't capture that, then we have no standard of care to compare to. So there's subtleties so, you know, CRS is a single construct, but there's subtleties within it that, you know, really inform the data we need to collect to get there.
Yeah, Michael, I'd actually, you know, even go a little step further, saying this is really a good kickoff to a call to action, is that we need more of that data generated, more option, more, you know, hospitals and pharmaceutical companies adopting this. It would be highly beneficial to the patients. And again, it's not just in-house or inpatient evidence and data that, you know, Cindy and Naz had spoken to, but actually going further out to the outpatient setting is really where this has the most promise, in my view, and the view of the discussions we've had throughout this CRS program.
Yeah, I would agree with that. I mean, what we're seeing from our customers is, you know, really the ability to get this into the outpatient setting is better for the patient. It's better from a cost perspective. It's better from a treatment perspective. But really, you need the tools to enable that to happen in the outpatient setting. One thing I would mention, and it's interesting, you know, from our clinical studies, we talk a lot about the outpatient setting. Unfortunately, what we've found with a lot of our clinical studies is there are some things that are broken in the inpatient setting as well. You know, we've been clinically shown to detect fevers related to CRS 4.5 hours earlier than the standard of care, and that was in the inpatient setting. So that obviously gets way worse in the outpatient setting, but I think these do have benefits to both inpatient and outpatient programs.
And I also would like to stress the importance of having diversity in these clinical trials for CRS, because we know that by nature, CRS can vary significantly across different demographics groups. We mentioned temperature, for example. But if we think of like how genetics can impact the immune system response to different therapies for CRS, or comorbidities, the prevalence of comorbidities such as hypertension or diabetes, cardiovascular disorders, that can also impact the timing and the severity of CRS. So having diverse patient population would ensure that the de-risking product can work across different demographics group. And in this sense, I think digital technologies are very helpful. As we mentioned earlier, they can help monitoring continuously vital signs at the comfort of patients' home. So they can reduce the frequency of in-clinic visits, and in this way, the trial can be more diverse and definitely more patient-centric, inclusive, and accessible.
Yeah, one quick point I wanted to carry on. Cindy brought up a really interesting point, was the issue of capacity with these new therapies. We started with CAR T therapies, and they're phenomenal. Bispecifics are coming like a freight train, and their universality of their treatment means there's a much bigger patient population, and CRS is as much of an issue with bispecifics as it is with CAR Ts, and I think DHTs really will help increase the capacity and the availability for those therapies.
Yeah, and I'll just add that I guess the current standard of care in the approved drugs is it's a little bit of a wild west out there. So I mean I think there's a very immediate there's a very immediate opportunity with DHTs to make monitoring more obviously more continuous and safer upfront. Like these algorithms do not need to be overly complicated to supplement what is there. So I think that it's DHTs can be here now. We don't necessarily need we don't need a deep neural network to save the day. So
Perfect. Thank you all for those thoughtful responses. Matt, you tied beautifully back into that immediate initial panels where we're thinking about how these tools can impact both inpatient and outpatient care. And Michael, you wrapped it up beautifully, given that call to action and moving us into our next panel. So we've discussed how important and critical it is for validating these CRS de-risking products, despite unique challenges that are faced throughout the field. But one thing remains clear is that clinical evidence still needs to be generated, and we have DHTs available now to get us into that path. So I would like to pass it over now to my colleague Ben, who's going to be moderating the next session, to focus on how we balance those clinical strategies as we consider the regulatory requirements when developing CRS de-risking products. Ben?
Thank you, Sam. And I'd like to welcome Chris from J&J and Seth from Bioformus and Actigraph to join me, please.
Good.
All right. I'm not seeing the spotlight, but I think we're good. So Chris, Seth, thank you very much for joining us. So in the previous session, we were focusing more on the product development side of things. You both have a regulatory background, and that's obviously very important if we're talking about building these kinds of products. So could you tell the audience a little more about what you believe developers of CRS de-risking products should focus on to ensure regulatory alignment as early as possible when they start designing and developing a product?
Sure, I can start. So first and foremost, just thank you for having me, and building on the previous panelists and all of the discussions, which are very, very centered around caring for the patients, both inpatient and outpatient. It all accumulates into the intended use, the indications for use, and the target performance that we're selecting and we're trying to substantiate where we're building these tools. And that is to be supported by a proposed analytical and clinical validation plan. So as you engage with competent authorities, you want to think about, how am I going to prove what we're building here? What are these keys to success? And what we've discovered, and through discussions with such said competent authorities recently, was by picking these time windows, as Michael had said earlier, right? There's 2 hours, 4 hours, 6 hours, defined grades of CRS, maybe 1, 2, 3, and then trying to figure out what's the scientific rationale behind that. How far away are the patients? How much need is there? How much risk is involved, really, is what we're hearing from the regulators, in the sense that Are there journal articles out there that can substantiate some of this selection of parameters that you're going to use? Are you talking to different key opinion leaders? Are you looking at other standards of care that you can draw corollaries from? And all while you really need to think about what are the human factors aspects that go into such considerations. So if we display an index or we display an output of a certain sort, what is that supposed to mean to a clinician who's caring for a patient who may be 2 hours away from the hospital? And how are they supposed to react? So you really need to think through all of these things before you approach. You want to have a good, clean, engaged discussion. And I think while it's good to solicit their opinions, coming prepared is absolutely necessary. I would not suggest anyone go in and just ask a blank question. Have a formed opinion first and foremost, and then work through that discussion with them as a collaborative partner. Chris?
Yeah, fully agree. And I think one other thing to think about is it's not necessarily just what and how the product work, but it's also how does it impact the downstream treatment and what are the ripple effects? So how does the product fit into the overall CRS management ecosystem? And what's going to be the impact by adding in this type of solution? Regulators are starting to consider about not just that box of the product, but how it fits in and what you need to do to demonstrate that it's not going to adversely affect both the treatment in a positive way, say for efficacy, but also is it not going to add any risks? And so I think, Seth, you mentioned that human factors protocol, and it's not just the how does someone interact with a product, but what happens next after a clinician uses it?
Maybe pulling on that thread a little bit, because a lot of the here initially we were focused on, you know, if you start thinking about your early interactions with regulators, what do you need? What homework do you have you done by then, ideally? But of course, over the lifetime of a product, there are many potential opportunities for engaging with regulators that go much beyond that initial design phase and where you're trying to put things together. So if you, from your personal experience, if you're thinking about what that could look like, that long-term engagement, like what are some tips, things that you've learned along the way that you feel are important to take into account?
Yeah, so I always think of it as a journey, right? And it's an appropriate analogy in the sense of you're starting off in the first engagement, and you'll probably have several sessions throughout the course until a submission is finally accomplished. But if you're thinking about multiple geographies, you're thinking about multiple standards out there, and I don't want to say there's different standards of care, but there might be different ways people interpret that standard of care. And you really need to seek a balance between how many patients you're going to strap a device on and, you know, the complexity, the cost, and the confidence that you need to really gain from studying all of this information. So part of that engagement is to find the boundaries and the limitations and or suggestions, if you will, from each of the competent authorities you intend to eventually place a marketing authorization into. Where do they draw those lines? And one of the reasons was the majority, I'll use a rough air quote there, of your patients have to come from our country or our market in order to substantiate. And that was really based on the concern that there are confounding factors built into these models that aren't so easily identified or transparent when we first engaged with them. And we learned over the time which of these variables and factors could actually sway the outcome. And so taking a conservative approach, sometimes you have to take into consideration how much data is going to be collected from each geography and which data can we justify, and really trying to think about their input as part of the input to the overall program is a key piece. I'll turn it over to Chris from there.
Yeah, and one thing that I always think about when you're starting to have those conversations is it's, you know, as a sponsor, right? You are living and breathing this technology, this device. You are so passionate. You've worked so hard, and You think you know everything. And this is where communication is so important, and with those early engagements, because you're essentially building your package that has to frame what your product is, sending it over, and hoping that there aren't too many miscommunications, right? Because there's always going to be this balance of how well do they understand your product, how well do you understand your product, and finding the right way to create that communication from the beginning is so important for building that foundation for those future engagements along the regulatory journey. So that's one area that I tend to focus on, especially in the beginning.
And Chris, would you say that if you apply what you just said across geographies, like Seth started to talk about, is there anything that you've learned along the way that has worked, that doesn't work, that are like important things to think about as early as possible if you have, as a company, that intent to go global?
Yeah, absolutely. And every country region has their unique regulatory components to consider. And it's not just those regulatory components, but it's also about patient management and how there might be unique aspects of patient management that are in different regions. And that's where it's really important to say, here's how my device fits in with the clinical workflow here, but then understanding that for each region that you might be going into. It's critical.
All right, thank you. Maybe to close us out with this panel, with a bit of a cliche question, but a very important one nevertheless. Like, if you could leave everyone with one take-home message, what would it be?
Yeah, from my perspective, is just believe in the process and the journey. It's there to help guide all of us along. I always think of the regulators as collaborators. They want the same thing. They're just trying to do the best for the patients, for the clinicians that are caring for these patients and loved ones. So I would never think of them as adversaries. We have to sit on the same side of the table. And when we engage with respect, really with our focus being that patient's journey in the end.
Yeah, fully echo that, Seth. And, this is where that early and often engagement is critical.
Good enough said about it myself. With that, thank you both so much. And I am going to hand things off to Jen Goldsack, CEO at Dime, to close us out.
Fantastic. Thanks, Ben, and congratulations to all of the project team on today's launch. These resources are so important for advancing the field, and I'm going to approach my closing remarks today perhaps a little bit differently than I would under other circumstances. I think many of you on the line will know that over the course of the winter break, I was diagnosed with cancer, stage 3C colon cancer. And since then, I spent 27 days in hospital. I'm happy to be home now. But I can tell you, as a cancer patient, being an inpatient is absolutely to the point where I am on the brink of being upset as I even reflect on it. No matter how good your nurses and your care team are, you don't want to be in the hospital. You want to be at home. And what you have done as a result of this project is you've opened the door for patients sort of pursuing IO therapies, whether as part of new medical product development or as part of routine care where those solutions exist. In the same way that we heard right during opening remarks, the ability for patients to be at home. And so the most important thing I need to do is thank you for your leadership and thank you for driving this forward. I can tell you firsthand, if there was an option for me to have gone home even one day earlier, I would have done anything. I would have been as compliant as you wanted me to. Whatever you needed, my answer would have been yes. So as I reflect on what you've achieved, I want not only to thank you from truly what is now new to me as a patient perspective, but also to sort of recognize where we are as we advance these sorts of technologies. We've learned today that we have to be really clear about how we define a context of use in order to use these new tools appropriately. We have to think about how we're educating not just patients like me to make sure that we are generating quality, trustworthy data that you can rely on for decision making, but we have to be able to support our clinician colleagues in being able to make sense of what these new streams of data actually mean so that they can have confidence sort of treating patients. And we have to reflect on not just how this is going to help sort of patients who have the option of being inpatient, but how we're now able to reach patients who never had inpatient, whether a clinical trial or sort of a prescribed IO therapy as an option for folks who may live too far away from a clinic, folks who couldn't take time away from their family responsibilities. We can now serve every single patient who could benefit from the development of new IO therapies or access to existing treatment options. And so this work is just so important. I also want to make sure that we don't lose momentum. The maturity of technologies, the sort of clinical decision-making, the scientific decision-making, the regulatory openness to these new streams of data to fulfill things like labeling obligations or IRB requirements for 4-hour or Q4 monitoring for safety. We know that we have these technologies at our fingertips. We know that we have thoughtful developers, every single one of our developer colleagues on this project team to start with. We know that we have the tools. We now need to come together and make sure that we are intentional and purposeful in driving the broad dissemination of these sorts of tools to do the greatest good. We not only have to serve patients, but we have to make it easier and more feasible for our pharma colleagues to actually be developing these IO therapies at pace, because we've reduced so much of the friction in the process. So Sam, I'm going to hand it back to you. Congratulations. Thank you for your hard work and leadership. And thank you to every single one of our partners who worked tirelessly over the last year to really move things forward. And I'll say just one more time, again, in a stance that's a little bit uncomfortable for me still as a patient, thank you. I can tell you firsthand the difference that you're making, and let's just continue to accelerate all of this work together.
Jen, thank you so much for those inspiring remarks. I'm trying to not to cry as well as we round out this session. It's so great to hear how these products can really lead to better improved patient outcomes, but not only the improved patient outcomes, but those extensions to those clinical trials, our pharmaceutical developers, and our clinicians as well. As we round out today, I want to invite you back to another session in 2 weeks where DIME launches our next project, Driving Innovation and Impact for Pediatric Care. We'll be releasing the new playbook for digital medicine for pediatrics, and this will provide solutions for the ability to meet the needs of children. And so we would like you to join us with that if you're able to join us in the next 2 weeks. Also, I'd like to invite you to join us on our next digital measurement project here at DIME, where we'll be focusing on how we can unlock sensor-based data to help optimize treatment for type 2 diabetes. So we'd love for you to partner with us on this journey as well. And with that, I would like to extend my sincere thanks to all of our panelists that have joined us here today for their valuable contributions. I'd also like to extend a thanks to our entire CRS project partner team for their hard work behind the scenes and getting us to this launch event today, as well as all of you for joining us today that are in the audience. Your participation and engagement has been truly appreciated. We want to thank you once again, and I wish you all a wonderful rest of your day. Goodbye.